Assalamualaikum and hi to all the lecturers and our fellow friends. Today our group going to present about the antimicrobial activity of date against the oral bacterial. This our group members consists of the YASMIN, me, SHAKIRA, FATHINI, and Siti Awatif. Next, we're going to move on to our objective for today. First, we're going to discuss the types of the dates. Second, we'll present the phytochemistry of dates. Third, present the antimicrobial activity of dates against the oral bacteria. And lastly is we're going to discuss the prevention of disease causes by the microorganism according to journals or literatures with the Islamic practices. Next, we're going to move into the introduction and dates as prophetic foods. You guys must be wondering, what is prophetic food? So prophetic food is a food that is mentioned in Al-Quran and recommended by our Prophet Muhammad sallallahu alaihi wasallam. So dates. What is the scientific name? So scientific name is a Phoenix dactylifera L, which is widely grown in Middle East and North Africa. Next, what are the common varieties of dates? Which are Ajwa, Medjool, Safawi, and Mabroum. Moving on to the nutritional composition of the dates. Dates have highly contained of carbohydrates like glucose and fructose. It is also have dietary fiber, vitamins like a vitamin A, B complex, vitamin C, and vitamin K. Other than that, it is also have minerals and bioactive compounds. All of these nutritional compounds can contribute to antioxidant, anti-inflammatory, and antimicrobial activities.
Now, I will be presenting about the phytochemistry of dates. Phytochemistry refers to the study of naturally occurring bioactive compounds that found in plants. Dates contain various phytochemicals that contribute to their antioxidant, antimicrobial, and also health promoting properties. First, dates are rich in phenolic compounds, which are among the most abundant phytochemicals that found in date fruits. This compound possess strong antioxidant properties and help neutralize free radicals and protect oral tissue from oxidative damage. This is important because oxidative stress has been associated with oral disease such as periodontitis and other inflammation conditions of the oral cavity. Therefore, phenolic compounds may help support oral health by reducing cellular damage and also inflammation. Next are flavonoids. Various flavonoids have been identified in dates, including apigenin. Apigenin has been shown to inhibit the growth and biofilm formation of Streptococcus mutans, which is one of the main bacteria that's responsible for dental caries. This suggests that flavonoids may play a role in preventing plaque accumulation and also promoting better oral health. Another important group of phytochemicals is tannin. Tannins contribute to both antimicrobial and antioxidant activities. In plants, they act as a natural defense mechanism against microorganisms. Similarly, in the oral cavity, their antimicrobial properties may help inhibit the growth of harmful microbes, while their antioxidant effect help protect oral tissue from damage. These also contain alkaloid, although in smaller amount compared to the phenolic compound and also flavonoids. Despite their lower concentration, alkaloid exhibit antimicrobial activity and have been reported to inhibit the growth of certain bacteria and fungi. This further contribute to the protective effect of date fruits. Another bioactive compound found in dates is saponins. Saponins can disrupt microbial cell membrane, causing leakage of cellular content and also leading to the bacterial cell death. Because of this mechanism, saponin contribute to the antimicrobial properties of dates. Lastly, dates also contain carotenoids. Carotenoids are natural pigment with antioxidant properties. They help protect cells from oxidative damage caused by free radicals and contribute to the overall antioxidant capacity of date fruit. This effect may help maintain the health of oral tissue.
Next, I will explain the three important oral bacteria that are commonly associated with oral disease. First, Streptococcus mutant. Streptococcus mutant is a gram-positive cocci that appears in chains. It is facultative anaerobe, which means it can be survived with or without the oxygen. This bacteria is the main initiator of dental caries. It ferments the dietary sugars, such as sucrose, to produce the acids. These acids will lower the pH in the mouth and cause demineralization of the enamel and will lead to the formation of the caries. In summary, Streptococcus mutans is the primary bacterium responsible for starting the tooth decay. Next, moving on to the Lactobacillus species. So, Lactobacillus species is a gram-positive roots. Unlike the Streptococcus mutans, Lactobacillus species is mainly involved in the progression of the dental caries rather than the initiation. They are acidic, which means they can survive and grow in highly acidic environment. These acidic conditions are often created by the Streptococcus mutans. Lactobacilli are commonly associated with the deep dentinal lesions and advanced caries. Therefore, Lactobacillus species contribute to the worsening and deepening of the caries lesion. Next, moving on to the third bacteria, which is the Porphyromonas gingivalis. Porphyromonas gingivalis is a gram-negative anaerobic root. It is considered as one of the major pathogens involved in the periodontitis. This bacteria produces several virulence factors that will damage the periodontal tissues. It also promotes the chronic inflammation of the gums and also the surrounding structures. Over the time, it can lead to alveolar bone loss, which may eventually result in tooth mobility and also the tooth loss. In short, Porphyromonas gingivalis plays a key role in development and progression of periodontal disease.
Now, I will present on how does the DIDs help in antibacterial action of oral bacteria. First is inhibition of bacterial adhesion. Before bacteria like Streptococcus mutans or Porphyromonas gingivalis can cause cavities or gum disease, they must stick to tooth surface or the gums. Phytochemical, especially tannins in dead extracts, bind to bacterial surface proteins such as fimbriae and adhesin. As a result, this coats the bacteria, masking their attachment sites. Unable to adhere to the acquired dental pellicle, the bacteria are simply washed away by saliva before they can colonize. This reduces plaque formation and also bacterial colonization. Second mechanism is disruption of biofilm production. Dead extracts contain bioactive compounds such as phenolics, flavonoids, and also tannins that interfere with quorum sensing. Quorum sensing is the process by which microbes communicate with each other inside a biofilm. As a result, bacteria cannot effectively communicate and coordinate biofilm development. In addition, dead extract can also inhibit the production of water-insoluble glucans. Without glucans, bacteria cannot firmly attach to tooth structure or build a strong plaque matrix. As a result, the biofilm becomes thinner, weaker, and less organized. Because the plaque structure is disrupted, it cannot mature into a harmful biofilm that causes dental caries and also periodontal disease. So, this is one of the mechanisms on how phytochemicals inhibit quorum sensing in biofilm production. They structurally mimic autoinducers and bind competitively to bacterial receptor proteins, effectively blocking the true signals that triggering gene expressions. Next mechanism is disruption of the bacterial cell membrane. Some bioactive compounds in dead extracts, particularly flavonoids that is hydrophobic, can directly interact with the lipid bilayer structure of the bacterial cell membrane and disrupt its normal structure. As the membrane becomes more permeable, essential cellular components such as ions and proteins leaks out, leading to loss of cellular homeostasis, resulting in loss of function and eventually cell death. Lastly, besides damaging the bacterial cell membrane, dead extracts can also interfere with the bacteria's internal machinery. Polyphenols can enter bacterial cells through compromised membranes and bind to essential enzymes, preventing them from functioning normally. In Streptococcus mutans, this reduces acid production, which is a major factor in dental caries. While in Porphyromonas gingivalis, dead compound can inhibit gingipains, which are virulence enzymes responsible for tissue destruction and disease progression. As a result, the bacteria cannot generate sufficient energy or maintain normal cellular functions, leading to metabolic failure. Next, I will move on to the evidence from scientific literature. A study has conducted that investigates the antibacterial activity of Finis dactyphilira or dead palm seed extracts against oral bacteria associated with dental caries, namely Streptococcus mutans, Lactobacillus casei, and also Staphylococcus aureus. The researchers prepared 2 dates extracts, which are aqueous extracts and ethanolic extracts. Antibacterial testing method used is agar well diffusion test that measure antibacterial activity by observing the inhibition zone around the well containing the extract. The larger inhibition zones indicate stronger antibacterial activity. As for the results, the study found that both aqueous and ethanolic date seed extracts inhibited bacterial growth. Ethanolic extracts show greater antibacterial activity than aqueous extracts. Lactobacillus casi was the most susceptible bacterium, while Streptococcus mutans and Staphylococcus aureus was also inhibited significantly. These findings indicate that date seed extract does possess significant antibacterial potential against oral pathogens involved in dental diseases.
Okay, next we will move to Islamic perspective on disease prevention. So, Islam encourages Muslims to maintain good health and prevent illness before it occurs. This can be achieved through healthy eating habits, personal cleanliness, and good oral hygiene practice. From a dental perspective, prevention is important because many oral diseases can be avoided through proper nutrition and oral care. One example of a prophetic food is the date or Phoenix dactylifera. Besides being a nutritious food, dates may also contribute to health promotions and also disease prevention, including oral health. To better understand this relationship, we will look at dates in Islam from a Naqli and Akli perspective. From the Islamic or Naqli perspective, dates are recognized as a prophetic food because they were regularly consumed by Prophet Muhammad SAW and are mentioned in the various Islamic traditions. This highlights their importance not only as a source of nutrition but also as a part of a healthy lifestyle encouraged in Islam. Prophet Muhammad SAW also said, "He who eats 7 ajwa dates every morning will not be affected by poison or magic on that day." These hadiths highlight the significance of dates in Islamic tradition. From the Akli or scientific perspective, contemporary studies have demonstrated that date seeds contain a variety of bioactive compounds, including tannins, phenolic compounds, and antioxidants. These compounds have been reported to exhibit antimicrobial activity against oral microorganisms associated with the development of dental caries. Okay, next, based on the antimicrobial properties identified in date seeds, there are several applications for promoting oral health and preventing oral diseases. The first application is the regular consumption of dates as a part of a balanced diet. For example, eating dates on breakfast, consuming ajwa dates daily, or breaking fast with dates during Ramadan. Dates contain naturally occurring bioactive compounds that may help support oral health by contributing to a healthier oral microbial environment. The second one is the use of date seed powder as a functional food ingredient. For example, it may be roasted, ground into powder, mixed with milk, smoothies, or yogurt. This allows the antimicrobial phytochemical from date seeds to be consumed easily, which can suppress the bacteria associated with the dental caries. Another application is the incorporation of date seed extract into oral healthcare products. Due to their antimicrobial properties, this extract may be potentially be formulated into oral care products such as herbal mouth rinse, dental gels, and natural antimicrobial formulations. By controlling the growth of carcinogenic microorganisms, this product may support prevention of dental and periodontal disease. However, further research is still required to establish in their effectiveness and suitability for routine clinical use. Overall, this application demonstrates how a natural product that is highly valued in Islam may also have a potential benefits in modern preventive dentistry. Although dates may contribute to oral disease prevention through their antimicrobial properties, maintaining oral health also requires good oral hygiene practice. Therefore, Islam promotes both healthy nutrition and good oral hygiene. Besides consuming dates as a prophetic food, Muslims are also encouraged to maintain oral cleanliness through practice such as using miswak, toothbrushing, and also using mouth cleansing. As mentioned by Prophet Muhammad SAW,
If I had not found it difficult for my followers, I would have ordered them to use the miswak before every prayer. Therefore, combining healthy dietary habits with proper oral hygiene practice may contribute to better oral health and disease prevention.
Okay, the presentation comes to an end. So the conclusion is dates have highly valued prophytic food. It contains various phytochemicals as we mentioned in the slide before, and mainly it is contributing to the antimicrobial against the bacteria, and dates also have excellent example of integration Islamic teachings and scientific evidence in promoting health and well-being. Thank you. That's all from us.